I mean, you were my student back at MIT when we started BE, and you were in 2109, which we'll sort of go back to, and part of the iGEM team. But what are you doing now? And yeah, what have you done, between MIT and now?
Okay, so yes, I'll start from the beginning. So in large part because of being BE as well as doing iGEM, obviously was interested in synthetic biology, wanted to continue that in grad school, and I went to Ling Chong Yu's lab down at Duke, and there I really studied sort of mathematical modeling of synthetic biological systems. So I felt, from iGEM and some of the other projects, we learned a lot of quantitative tools, but we weren't really applying them as much. So, basically trying to uh, mathematical model different spatial patterns, and was able to engineer a system of positive and negative feedback loops with diffusible molecules to make patterns in space, and developed a stochastic agent-based model for that. So that was pretty cool.
Yeah, that's like a goal, right? Like that's been one of those things that people have been working towards. It's really hard.
Oh yeah, like I was in my third or fourth year, and I was like, maybe more like second or third, and I was just like, I don't know if this is gonna work. Like, doesn't seem like it's going that well. And then I made some tweaks to the system and basically started to see, different phases occur in a plane, and then that gave me hope. And then I kind of optimized the system around it to get it to go, but definitely not the easiest project I was a part of.
No, very, very tough. So, so you're not at Duke anymore, though, right? You've left that area.
Yeah, so I graduated in 2013, and went back up to MIT, and I think we met up once when I was there with Ron Weiss's lab, and one of the impetuses really was, I love the pattern formation project. It was cool, but practical applications, you kind of have to stretch, a little bit. So it worked with Ron Weiss on RNA circuits to help differentiate stem cells into beta cells, and the technology was pretty cool. Actually, I think a lot of the technology ended up getting spun out into Strand Therapeutics. Not sure if you're familiar with them, but
I am.
Yeah, they're doing mRNA, and I think they're focusing on vaccines now. But
who ever heard of an mRNA vaccine?
So my job was basically to get self-replicating RNA into iPS cells, because direct genomic DNA modifications often get silenced in iPS cells. So I did some microRNA switching. The idea was basically let's switch on at the right time to give whatever factor to move to the next thing. I wasn't there too long. I knew I wanted to get into industry, and I definitely like Ron Weiss, and
interesting project, learn a lot, and then when a nice opportunity to join industry comes along, I think as a postdoc, that's the right next step. So awesome. Yeah.
So I went to Greenlight Biosciences, which they've done pretty well lately. I know they actually put a satellite site down here in Raleigh, Durham, but was part of an ARPA-E project to basically start with methane and go to isobutanol. And it basically, one of the advantages of the technology was it was all cell-free. So you could mix different extracts from different organisms. And so I did a lot of work with methanotrophs and a lot of work with E. coli to sort of, the idea was, let's take methane, make it to methanol, make it to formaldehyde with the methanotroph extract. And with the E. coli extract, you can go all the way to isobutanol.
That's so interesting. I've never really thought through that notion of sort of staging the extracts so that the circuit would perform in series, right? And that's very cool. Very cool.
Yeah, it was really cool. It definitely learned a lot, and you know, unfortunately, you know, didn't really scale.
That's the thing about extracts. You know, biology is amazing because it self-replicates, and then when you start to go to extracts, you're like, well, I'm taking away one of the really strong things about using biology. Right?
Yeah, especially when you're talking about a pathway that long, you know. So, you know, actually, we went to there was a conference that. Because they gave out, like, it was ARPA-E, they gave out like 30 of these grants to different academics and industry. And I think most people, the consensus was it wasn't going to scale. So unfortunately, they laid off our whole team. But, you know, that's how it is.
That is, yep. Yep.
And so, you know, applied to a lot of jobs from there. And all of the jobs ended up being in San Francisco. So I was like, guess I'm moving there. And, you know, at this time, actually, the timing worked out pretty well because I got engaged. I got married right before then. My wife had just finished her PhD at Duke. So we were doing long distance from Boston to Duke for like, what was it, a year or two? And, you know, so that was a good thing. And, you know, so she was looking for opportunities too. And so she got a job doing technical writing at Thermo Fisher in the Bay. Nice. And because she's got a weird story too. She got her journalism degree. Oh, that's a nice looking cat.
That's Milkshake. We got her the year that I taught you, actually. So she's an old lady at this point.
Oh, cool. Yeah. And, you know, then she read about the 1918 influenza virus and went back to school and got a biology degree and then did her PhD in virology. So yeah,
so she's kind of busy now too, right?
So she was writing technical manuals for all of the COVID testing that Thermo Fisher was doing, and she was quite busy.
Yes, what a story! What a story! And you have a child at this point, so
yeah, so. Yeah, I went to San Francisco. I took the job with REG Life Sciences, and I can't say enough good things about the people I worked with there. They were just so friendly, and it's a laid back. I felt like maybe we didn't work as much, but we worked more efficiently. We had just a great infrastructure in place because they were LS9 for. Right. You know, they had a long time to figure it out.
Figure out how to work. Yep.
So I worked on, you know, a medium chain fatty acid that we are commercializing in Europe. We scaled it to 130,000 liter scale. So that was cool. Then I was kind of a team lead managing some research associates as well as you know, coordinating among metabolic engineering, high throughput screening and fermentation to do an ExxonMobil partnership to make biodiesel.
Oh my goodness.
The project was pretty cool because I was a sugar utilization lead to sort of get alternative sugars used as well as glucose and at the same time. And we were doing.
Can I just stop you there? You choose very hard projects.
Well, yeah, that one, I still say that one was probably easier than the other two, the Weiss project and the other one. But it was still, yeah, it was good. So I saw just the other day they published a patent on it. So that was good to see.
That is great.
And but a little disillusioned working for ExxonMobil. So after I left, they were acquired by Genomatica, and then I think later on when COVID hit, ExxonMobil was like, "You guys reached all your goals, but we're not going to do anything with it right now."
Right. We got other fish to fry.
Yeah. So, I left, and some of my colleagues said I was a little crazy, I guess. I left there to start a company. Yeah, you mentioned my son. I need to bring up my son. Right. I think about 6 months before I did that, my son was born. So in March of 2018, and
congrats.
Yeah, he's the best little guy. He's very sassy. He's running around. He's talking a lot. He just won't, like, we were waiting for him to talk, and then once he started talking, he just won't stop.
Here's what I think.
So he's been cool. We're around 1 year old. We moved back here. So started the company to I saw and this was even before a lot of the people were getting a lot of money for it, but I did see there were some people trying to make glucose to cannabinoids, and actually this might be where I got lazy, I guess. I was like, why are you trying to do that whole pathway? It's a very complicated pathway. So, our idea was essentially let's start with low value cannabinoids, abundant ones, and just do 1 or 2 step conversions.
Right. Of the Bioengineering.
So, I was doing a cell-free extract to do one of those conversions. But it could also be applied to synthetic chemistry. So after 6 months in the Bay and realizing it's extremely difficult to operate a company there without a lot of money.
Right.
I moved down to Durham. Since we were familiar with the area, but also, there's a lot of pharma here. There's also a lot of ag tech. So, and I thought the technology really went hand in hand with ag tech.
Yeah.
Because, there are certain cannabinoids we avoided where we knew the plant biologists are gonna totally kick the synthetic biologist's ass. So
let's not do those. Let's leave those for them.
Yeah. So I hooked up with my buddy that he was getting his PhD at Duke at the same time in organic chemistry, and spent some time in pharma. And we developed a process to go CBD to CBN in a synthetic chemistry route, and it was really good process. We've scaled it to 10 kilograms per month. And we were going pretty good on the revenue in terms of, because you could sell these at like 10K per kilo. So that's kind of what we're up to there. We're looking at cell-free extracts to make, some of these conversions happen, but also using synthetic chemistry as well.
So exciting. When I hear all that you've done, it just strikes me that the ability to integrate so many different aspects of science, like we so artificially segregate the way we teach and learn science, and everything you have done has really integrated all, they call it STEM, right? But all of the technologies like the cell-free extracts and the engineering in order to like mix and match and optimize output. And then, you've got the science, the math, you've got the mathematical modeling, you've got the chemistry, the biology, like you are like, you should just have STEM as a tattoo. Like you really bring it all together.
It's it's exciting. I mean, I know somebody once gave me the advice that I should that being what do you call it, an expert in a very small specialization is a good thing to do. And I've kind of completely ignored that advice, and I don't know if that's a good thing or a bad thing, but it certainly seems like, looking around, it seems like a lot of people are looking for people that can integrate the skills across things. I mean, if you pick the right specialty, it's great, but everything's evolving so fast that, and it's too late now. So,
well, you have clearly made it work. And I think that that's amazing. It's a wonderful career trajectory. I think you're doing amazing stuff. I'm sure it's going to go, off the charts. It's going to be amazing. So that's going to be really incredible to watch. I'm so excited for you.
Yeah, I'm excited about what the future has in store, and